Okay, well, stop me if you can. I can't really see the chat. But hi everyone. My name is Aditya Srinivasan, and I'm a senior from San Ramon, California. And today I'll be talking to you about my project on mRNA vaccines and their role in treating cancer. Immunotherapeutic treatments for cancer, or using the body's immune system to attack its own cancer, is a topic I'm quite passionate about, and my time here in Polygence culminated in me writing a review paper on this topic. First off, I think that it is important to address the elephant in the room, the pandemic around us. Synthetic mRNA vaccines are a relatively new technology, and the Pfizer-BioNTech COVID-19 vaccine is actually the first one to have been approved by the FDA. This technology has truly blossomed due to the pandemic and has been placed front and center in the court of public opinion. Misinformation about these vaccines has been rampant and has had major life-changing consequences. Learning about them has become an urgent public health issue, and that was my inspiration for pursuing this project. I hope that through this presentation, I can spread accurate and useful information about mRNA vaccines for your benefit. Before we move on, a fun fact is that the reason mRNA COVID vaccines were able to be created so quickly is due to researchers who are developing mRNA-based vaccine strategies for cancer, adapting their technologies to tackle SARS-CoV-2. Before we get into specifics, everyone must have a solid understanding of what vaccines are and how they work. Vaccines are a way to prime your immune system and help them develop defenses to diseases they have not yet encountered in the real world. An antigen associated with the disease is presented to cells, which activates a cell-mediated immune response involving cells like helper CD4, and cytotoxic CD8 T cells. CD4 T cells also activate B cells, part of the humoral response. B cells produce antibodies to neutralize the antigens, and memory B cells are stored in lymph nodes to provide a rapid and specific response in the case of future infections. Cytotoxic CD8 T cells, also activated by helper CD4 T cells, attack and kill cells carrying or infected by the antigen, combating sickness. That was a lot of words. Let's take a look at a graphic. Here you can see the vaccine leading to the activation of CD4, CD8, and B cells. Another relevant topic is the central dogma of biology, which states how the DNA inside our cells is transcribed to create mRNA and how that mRNA is then translated to create proteins that control our bodily traits and functions. This will be important in just a minute, so hold on to it. This graphic here, it specifically refers to mRNA vaccines, which is why the first half looks a little weird, but we'll get back to that. This actually leads straight into my next slide. How do mRNA vaccines work specifically? All cells carry a variety of surface proteins that are vital to their functioning. Cancer cells, however, are heavily mutated, which leads to them carrying two notable types of surface proteins, or antigens. Tumor-associated antigens are antigens found on most cells, but they're more prevalent on tumor cells. And neoantigens, which are tumor-specific, are only found on tumor cells. Researchers synthetically create strands of mRNA that code for these antigens by sampling the patient's tumor cells. Each strand of mRNA can only code for one antigen, but a single vaccine can carry many strands, which is useful because it provides the immune system with more identifiers for the diseased cells. These strands are often chemically modified to increase stability and translational efficiency. They're also delivered in vectors, like lipoplex nanoparticles, to increase cellular uptake. The SARS-CoV-2 mRNA vaccine uses lipoplex nanoparticle vectors, which is why they have to be stored in cool temperatures. Vaccines must also contain a delivery solution and an adjuvant that helps activate the immune system. Once it has entered the cell, the vector releases the mRNA, which is then translated multiple times by ribosomes to create the antigens that will be presented by the cell to alert the immune system. The immune system, if properly activated, will then attack all the diseased cells. Here you can see a graphic of how this whole process happens. If you look at the right side, you can see the antigen-coding mRNA in a liposomal nanoparticle vector. And going back to our first graphic, the nanoparticle is the circular object on the left side. Cancer vaccines use mRNA that code for tumor-associated antigens or neoantigens, while the mRNA vaccine for COVID would code for a piece of the virus's spike protein. I'll stay here for just a second because there's a lot going on here. This leads to the question, can this be done with different types of vaccines? And the answer is absolutely. Once again, the only objective is to get these antigens into the cell and into the view of the immune system. This can be done with peptide vaccines, which are very common and involve just injecting the antigen itself. The flu vaccine, for example, is a peptide-based vaccine. DNA vaccines work very similarly to mRNA vaccines, and going back to the central dogma, they just require the extra tedious step of entering the nucleus and being transcribed into mRNA. The AstraZeneca and Johnson & Johnson vaccines are both DNA-based. Whole cell vaccines use an irradiated tumor cell itself, carrying all its characteristic antigens in the vaccine. Each of these modalities has its own benefits and drawbacks, which I, unfortunately, don't have much time to get into here, but they boil down to DNA being a more stable molecule than RNA and whole cell vaccines providing a more robust immune response. I'm glad to answer any questions about these modalities after my presentation. And mRNA vaccines themselves are advantageous for many reasons. Since they only code for one or a few antigens, they trigger a specific immune response that minimizes side effects. Compare this to current cancer treatments like chemotherapy, which attack any rapidly multiplying cell and therefore have a plethora of painful side effects. You can also safely receive many doses of mRNA vaccines without any adverse effects because mRNA is naturally degraded by the body. It simply doesn't stick around long enough to cause any serious side effects. This information is especially pertinent to those of you looking at booster shots right now, but in terms of cancer, multiple doses can similarly help bolster the immune attack against tumor cells. mRNA vaccines also never enter the nucleus of your cells like DNA-based gene therapies do, and so they don't pose the risk of insertional mutagenesis, which is the integration of foreign DNA into your own. At this time, I'd like to pause for a quick second and make a PSA. I said a while ago that the AstraZeneca and Johnson & Johnson vaccines are both DNA-based, but I want to make clear that they do not pose the risk of insertional mutagenesis. This is because the vector that these vaccines use to deliver the DNA is an adenovirus, which simply does not have the machinery to integrate the DNA into your genome. So if you receive the J&J or AstraZeneca vaccines, you can rest easy. mRNA vaccines are also cheap to produce and can be made very quickly once the genetic sequence of the target antigen is discovered. This is especially useful in time or economically sensitive situations like the coronavirus pandemic and hints at a future of cancer treatment that doesn't bankrupt its recipients. Finally, both mRNA and DNA-based vaccines initiate a cell-mediated and humoral immune response, like I mentioned at the beginning of this presentation, and this makes them more likely to overcome vaccine resistance than other modalities. And like I said, while peptide-based vaccines have been around for a long time, nucleic acid vaccines are a relatively new technology, and the pandemic has played a large part in giving them the attention they need to grow. You might wonder why, after all these advantages, mRNA vaccines aren't more prevalent. There's three main reasons why. Firstly, there are long-standing prejudices in the scientific community against using mRNA as a vaccine material because of its instability. Secondly, foreign nucleic acids have a high immunogenicity, meaning that they attract the immune system much more easily than other pathogens. This is a problem because if the immune system attacks and destroys the mRNA before it reaches a cell and has a chance to be translated, then the vaccine will not work. And finally, due to a variety of biological factors ranging from difficulty with priming immune cells to difficulty penetrating the immunosuppressive tumor microenvironment, mRNA cancer vaccines are just often not very effective. But recent innovations by researchers have addressed many of these problems. Biochemists Carrico and Weissman, both at the forefront of mRNA research, found that modifying one of RNA's bases, uridine, to make pseudouridine, reduced its ability to trigger toll-like receptors and initiate a misguided immune response against the RNA itself. It also increases the stability of the mRNA molecule and its translation rate. Different vaccine formulations, such as using different adjuvants and using different delivery methods or vectors, have all been shown to increase the vaccine's effectiveness. And finally, the creation of new mRNA constructs, such as self-amplifying RNA and trans-amplifying RNA, which can propagate themselves within the cell and thereby create more antigens for the immune system to notice and attack, also helps make mRNA vaccines more effective and a more promising modality for the future. With the FDA recently approving their first IVT mRNA vaccine, the future is brighter than ever for this modality. New innovations, like I just mentioned, increased funding due to the publicity received during the COVID pandemic, and combination therapies, which help increase effectiveness, all ensure that mRNA vaccines' prominence in the medical field will only grow. Thank you so much for listening, and at this moment, I'd like to take a moment to thank my mentor, Alexandra. She has been an invaluable source of knowledge and advice throughout this whole project and helped me decide on, create, and perfect my paper. I truly could not have done this without her. Thank you for listening, and I'm glad to take any questions you might have.
Hi, everyone. Sorry, I'm a bit late. My previous session ran over. My name is Ali Munson, and I am going to be moderating the rest of this session. Thank you so much, Aditii, for your presentation. That was really great. I came in a little bit late, but from what I heard, it was quite impressive. So yes, if anybody wants to type questions in the Q&A section under the session tab or the chat, or you can also request to share audio and video, and you can also ask live. I'll go ahead and start off with a little, oh, we got one, okay. Ashley asked, do you think mRNA vaccines will be prone to resist mechanisms seen with other immune approaches?
Well, yeah, mRNA vaccines, they still have a very long way to go before they become like a truly effective modality. I didn't get into it too much in this presentation, but they face a lot of issues with penetrating the tumor microenvironment and also priming CD4 and CD8 T cells well enough to create a robust immune response. So they are subject to those difficulties right now, but the thing is, the only direction is up, so future innovation stuff will only help address those difficulties and make our mRNA vaccines a more effective and useful modality in the future.
We'll give it a few more seconds. Alrighty, well, thank you so much, Aditii, for your presentation. And next presenting will be Katie, who is a junior from Danville, California. Oops, put one too many people up.
Hi, can you hear me?
Yes.
Perfect. And then let me share screen. 1 second. Okay, wait. Sorry, let me go here. Okay, can you see everything or?
Yes, I can see your presentation.
Okay, perfect. So I'm Katie Katerndahl, and I'm going to be presenting on catecholamines and anxiety diagnosis. So the prevalence of anxiety disorders as a devastating psychiatric disorder is exponentially rising in the modern general population. Currently, the predominant methods for diagnosing anxiety disorders are through self-report questionnaires past childhood or beyond. but unfortunately, precise data is difficult to obtain as individual responses on anxiety questionnaires vary drastically. So, abnormal catecholamine levels may be a strong indicator of anxiety. By investigating a potential correlation between anxiety and catecholamine levels, anxiety can be addressed at an earlier stage, allowing for reduced variability in qualitative responses and ensuring proper diagnosis in children, adolescents, and adults. So, catecholamines, including dopamine, norepinephrine, and epinephrine, modulate stress-induced activity. The neurotransmitter dopamine influences a person's emotional responses and mood, sleep, memory, and have been associated with the development of anxiety through abnormal levels. Norepinephrine is an excitatory neurotransmitter and stress hormone associated with the fight or flight response and increases alertness, attention, heart rate, and anxiousness. And epinephrine acts as a neurotransmitter and hormone in which the activation causes elevated heart rate, muscle strength, blood pressure, and anxiety symptoms. So, the prevalence of adolescent and adult anxiety disorders continue to rise from previous generations. Children diagnosed with anxiety aged 6 to 17 years increased from 5.5% in 2007 to 6.4% in 2011-2012. Young adults increased from 7.97% in 2008 to 14.66% in 2018. So it's an even more pronounced increase. So to diagnose an individual with anxiety, at least 3 of 6 symptoms, such as restlessness, fatigability, difficulty concentrating, muscle tension, irritability, and sleep disturbance, have to be present for a duration of 6 months or longer. So currently, the predominant methods for diagnosing anxiety disorders are through parent report or self-report questionnaires in young adults and older generations and children for parent reports. So the preferred method of testing in the US is the Diagnostic Statistical Manual of Mental Disorders, the 5th edition, or the DSM-5. However, a limitation of this is inherent biases that exist due to methods of testing, including the possibility of over-reporting. Also, the individual's interpretation of the language used could potentially influence the results. So, in order to address these inconsistencies, a more quantitative method would be beneficial to help misdiagnosis of anxiety disorders. So, a potential biomarker may provide insight into early detection. So, the procedure for completing this review involved looking at 15 of the leading studies: subject, hypothesis, procedure, including type of catecholamine analysis, so plasma or urinary, average age, grouping, and the results. So all 15 had common characteristics, namely each subject underwent a blood or urine test before and after the experiment to determine levels of biochemical assays, and also participants completed an anxiety test, most typically the DSM-5, and each study measured catecholamines in anxiety disorder patients and control group. So these studies were based on the effects of catecholamines on anxiety-related behavior, and the patients were found to have significantly higher levels of epinephrine, norepinephrine, and dopamine levels as compared with the controls. So performing a meta-analysis of publicly accessible data, a direct correlation between catecholamine levels and anxiety disorder was found by analyzing pre-experiment baseline values and anxiety scores. So with the provided sample sizes of any pre-experiment measurements of catecholamine levels, the means and standard deviations of the pool groups were compared using two-tailed unpaired t-tests, assuming Guyson distribution, with both populations showing the same standard deviation. So this is when the unpaired t-method tests the null hypothesis that the population means related to 2 independent random samples form an approximately normal distribution. So the data sets were then analyzed using graph pad analysis and used for statistical testing, and statistical significance was found between select control and anxiety groups. So the data analyzed included any pre-experiment baseline measurements of epinephrine, norepinephrine, and dopamine. So only which then measured using urinary and plasma enzyme-linked immunoabsorbent assays, or ELISA analysis, which is used for determining the concentration of target protein in biological samples. So only data that compared anxiety patients with a control group of subjects that had undergone experimental treatment were used in order to ensure there was no contributing factors that could negatively impact the results. So summarized pre-experiment urinary epinephrine values report an overall two-tailed unpaired t-test of 0.1672 and a paired t-test of 0.1144. Norepinephrine found a two-tailed unpaired t-test of 0.0778 and a paired t-test of 0.0577, and dopamine urinary values found significance in unpaired and paired of 0.0316 and 0.0426. So, when plasma values of the same studies, summarized pre-experiment plasma epinephrine values report an overall two-tailed unpaired t-test of 0.0100 and a two-tailed paired t-test of 0.0082, which is significant in both. And then for norepinephrine, unpaired found 0.5673 and paired found 0.3123. So two-tailed unpaired t-test of urinary epinephrine summarizing 7 studies produced no significance when analyzed, and two-tailed unpaired t-test of plasma epinephrine summarizing 3 studies produced statistical significance. So in urinary, there was no significance, and same with plasma, but in dopamine urinary values, there was significance in both. So these findings reflect the need for recognition and treatment early on. By identifying early indicators of anxiety disorders, patients can be more efficiently and accurately identified for diagnosis and given appropriate treatment to address their condition. Utilizing non-invasive assays such as urinary and plasma catecholamine analysis can give insight into early diagnosis and treatment for a multitude of anxiety disorders. Quantitative testing removes the subjectivity and reporter bias. So through a deeper understanding of these biomarkers and other potential comorbidities, there is hope to reduce the risk of suicidal ideation through early recognition and treatment of this crippling condition. Thank you.
Thank you, Katie. Great presentation. We're going to open up the floor now to any questions.
Sure, and then let me stop sharing.
I'm kind of curious, do you happen to know if the urine versus plasma tests, if one or the other is more reliable? It was kind of interesting to me to see the different trends between the two.
So from what I've seen, urinary analysis is actually more reliable in that the each method used is kind of more consistent than the plasma. However, I think that a study should be made where you test the two to see them, because it wasn't complete. Both methods are still used.
Interesting.
Does that answer your question? Or?
Yeah. Sorry, I'm just kind of puzzled by it. I know, one of the biggest roadblocks, I majored in neuroscience, and I'm studying it now. One of the major roadblocks in studying a lot of disorders and diseases in the brain in humans is methodology. A lot of animal studies are being done, and so I'm always just kind of curious and perplexed as to, how can we really translate this to humans in order to, as your presentation told us so eloquently, screen and be able to kind of help diagnose people with different mental health disorders as well.
Yeah. the studies were definitely limited within the time period. So I think more research definitely needs to be done there.
Yeah, and I'm curious, did you come upon any papers that might have done maybe like fMRI in conjunction with?
Some, but mainly they were with different mental disorders other than anxiety, so they weren't as relevant.
Yeah, and I wonder if, one day they'll be able to come up with some better like injection biomarkers in which, we could maybe like tag directly for these molecules like dopamine and whatnot and be able to actually see their expression in the brain through maybe MRI or fMRI or something. Very complex, but maybe one day we'll get there. Alrighty, well, thank you so much. I don't see any other questions as of right now, but if anybody does have more, feel free to put them in the chat, and students can answer your questions accordingly in there. But thank you, Katie. We're gonna move on now to, let's see if I can do this correctly. Yes, okay. Next we have Teesha Sreeram, a senior from San Jose, California.
Hello.
Hi.
All right, let me share. All right, hopefully you guys can see my presentation.
Yes.
All right. Hi, everyone. My name is Teesha, and this is my presentation on my Dimensions of Health Toolkit. So just a little brief agenda of what I'll be talking about today. I'll start off with a little introduction of myself and my project, some goals for my project, my current outcomes, and the next steps for my project. So once again, hi, my name is Teesha Sreeram. I'm a senior from San Jose, California, and my interest in public health stemmed from a class called Contemporary Health Concerns. And in this class, I learned all about the 6 dimensions of health and their applications in real life. At the end of this class, I created a PSA about prediabetes, and that got me interested in this format of providing information. So that gave me the inspiration to create a toolkit in this way. So now I'm going to talk a little bit about what the 6 dimensions of health are. So they're listed here on the screen, and briefly about each of them. Physical health has to do with anything with your body. So like, eating habits, sleeping habits, exercise habits. Emotional health is directly tied to mental health and mental well-being. So like in dealing with emotions and stress, etc. Environmental health has to do with the environment around you, such as your workplace or your study area, but also outside of that, like community environments, such as, clean air, clean water, etc. Spiritual health has to do with your personal moral values. Intellectual health has to do with constantly challenging yourself and growing your intellectual base. So, studying more, learning more, etc. And occupational health has to do with anything related to something that will help you with your occupation. So interview skills, job skills, etc. And so what is my project? So looking here at this graphic, these are the 6 dimensions of health once again, and it is a toolkit surrounding this. So my intended audience is high schoolers and young adults, and I want this to be an informative toolkit to leave readers with ideas and resources of what health is and how they can further their health journey. So it starts off with information and then some things that they can take away from the toolkit. So here's like an example layout from my toolkit. It starts off with a case study about one dimension, and then it follows that up with an activity for personal improvement, a PSA, and further resources. So this is like the format used 6 different times for each of the 6 dimensions. So taking like physical health as an example, start off with a case study, a real world application of physical health, and a reflection piece / activity for personal improvement where one can really engage with it on a personal level and kind of identify what their goals are for that dimension. And then a public service announcement, so just a different format of providing information to the reader and leaving them with further resources that they can use to work on their goals and find more information about any topic that interests them in relation to the toolkit. So current outcomes of my project, I have a toolkit in the final stages, and I'm in the process of distributing it within my personal network to get it out to as many people as possible. So for the future, I hope to reach out to more people to publicize the toolkit, and I want to create some next steps outside of the toolkit. So toolkit is just a very basic starting point, informational. I want more steps after that. So creating a community center or a program that people can attend to learn more about health on a personal level, because this way they have more resources to engage with to kind of help themselves in their health journey. So here's a couple of examples for next steps, looking at emotional health, working with schools to provide more resources for their students in relation to mental health. Sometimes school can get really stressful, so just listening to the students and kind of working with administration and kind of, bettering their existing resources or creating new resources to kind of, engage with the students more in terms of emotional health. And then looking at physical health, as I mentioned earlier, pre-diabetes was kind of one of the starting topics for this toolkit. So looking at a reversal program for teens in conjunction with local health systems, just like more community-based ways that people can get help so that we can prevent worsening health in society overall. And then just once again, to summarize, my project is a single toolkit around the 6 dimensions of health with activities and resources that one can engage with. Here's my email if you guys have any questions. Thank you for listening.
Thank you so much, Teesha. We're going to go ahead and once again open it up for questions. I was just kind of curious, for the various case studies that you do you hope to or attempt to make them rather, I don't want to say unique, but maybe diverse? Because I have a feeling, in order to appropriately reach different communities and make it kind of widespread, that might be kind of a good tactic to pursue.
Yeah, so when I kind of took inspiration from materials that I saw in my contemporary health concerns class, and I kind of created the toolkits in that way. And so I didn't make each of them very, very specific, in conjunction to a certain specific physical health problem. I kind of made it here's a more general issue with physical health and how here are some ways that you can go about it. So yeah, I didn't want to make it super specific, because then I feel that would limit the kind of understanding of it. So yeah, I did decide to go with a little more general overview when creating those case studies.
Gotcha. Nice. One question here is, for your toolkit, do you think accessibility may be an issue?
That is an interesting question. I do see where you're coming from. I think because I'm still looking at ways to put it out there,
I feel like yet to overcome this, but like, you know, like looking in the future, I can see how it could be hard because right now I'm like mainly going through social media and personal networks, and not everyone has access to that. So I think some other ways that I was thinking of overcoming that barrier is in the area that I live, we have a lot of community-based clinics, you know, perhaps like reaching out to them and saying like, hey, can I put like a QR code up here? Can I print out some copies and like put them in your waiting rooms? So that way, you know, people who do enter those spaces do have access to that. I think that's kind of one way. Another way is like, you know, just like looking at places around me. So for example, our library has an area where you can put like advertisements up, putting it there. So I'm kind of trying to identify places where I know like a lot of people will kind of cycle through and focus on those areas to put out more hard copies. And then in terms of social media, just getting it out there, and then hopefully, you know, people can like send it to their friends from there, etc. So yeah, I think in terms of accessibility specifically, perhaps like, you know, once I get it out to more people, if I'm seeing that people are enjoying it and wanting to engage with it more, looking into translating it to different languages, you know, specific to my area, and then kind of going from there. And I think identifying different ways. That's one of the reasons I chose to do a PSA, because perhaps, you know, a video could be a little more helpful to certain audiences over reading a toolkit. And that's where my next steps also come in. I think that will help with accessibility, is if they have a more like, almost like more like of a physical place to go over just reading a toolkit to kind of receive more help.
Amazing. Thank you so much for your presentation and for your answers. Next up, we are going to have Aditya, a senior from Fremont, California.
All right, you guys can see me, right? Or just not working. Can you hear me as well?
Oh, that's a lot better. There we go.
Okay. This screen. You can see the presentation, right?
Yes.
So hello, my name is Aditya Mangalampalli. I am a senior from Mission San Jose High School in Fremont, California. And our project is about tumor localization and monitoring tumors using haptic pressure data. I personally have been fortunate enough not to know anyone, or not anyone in my immediate family who has been diagnosed with cancer, but I do have friends who have relatives or whose personal family members have gone through it themselves, and that kind of really inspired me to pursue this a bit further to see what else I could do to improve the space that it's currently in. So some general info about cancer is that cancer is currently found in the form of tumors, which are these large lumps of cells. So you can see on the second picture here. These are the normal cells, and they're just tumor cells that explode out of it. And basically, late treatment can cause this cancer to spread to other parts of the body. And when it does that, it can block arteries and major blood vessels, and it can lead up to death. And cancer currently affects around 18 million people worldwide. And statistics do show that 1 in 3 people will get diagnosed with cancer, and 9.5 million people die each year due to this cancer. On the right-hand side, you can see a diagram of the different types of cancers and how they've just been increasing over the past few years. And on the left-hand side, you can see the difference in diagnosis. As you can see, the U.S. usually diagnoses, and U.S. and Australia usually diagnose the most because they have more developed healthcare systems. But the rest of these countries don't actually diagnose them because they don't have the facilities or the machinery required. So tumors are currently detected through CT scans or MRI scannings, and these scans are very time-consuming, 30 minutes for a scan and 24 hours for a result for a CT, and 90 minutes per scan and 24 hours for a result for an MRI. And they're very expensive, $3,000 for a CT, MRI $6,000, and to buy a machine or to start a hospital, it can range anywhere from $2.5 to $3 million, which is, on the scale of some developing countries, not very affordable at all. So due to the severe nature of cancer and how time critical it is, mainly due to the fact that these cancers can spread within like days, more affordable and faster detection options are currently required. It needs to be accurate, affordable, and very quick. So the proposed solution is to take advantage of different pressure sensors and pressure resistive values at different points of a tumor to localize points of interest and then plot them to make it easy for anyone to see and to have immediate reporting of results. So in terms of the hardware, the device will sample pressure at various points of an organ, and its movement will be tracked through localization algorithms, and it will be using a pressure sensor to read the values and then chart them to see where there's outliers and anomalies to determine if there's a tumor or any hard surface that's unnatural in a particular given area. So on the left-hand side, you can see the Naruko cube. A Naruko is called a fiducial, which basically means it's these really big barcode things that are really easy for a camera to detect, since they have a lot of contrast between the background, the white border, and them themselves. So this was called a fiducial, and this was mainly what was used in order to track the stylus when it was plotting values and collecting data. And currently it's a cube, so all 6 sides have a different fiducial side, only 3 are shown here. But what this allows us to do is, even if the cube was rotated or like pointed at a different angle, some part of the cube would still get captured, and it would basically be able to the camera would be able to figure out exactly where the cube is in a relative position to where the camera is. On the right-hand side, you can see the circuit that was used. The basic bare circuit that was used to send data to the computer or the other device that's plotting the data. So in this case, we have a Node MCU ESP8266, which is a microcontroller. And it's a simple button circuit. So all it does is it pushes a button, it can read that value and send it. There is a pressure sensor attached. It's not shown because the pressure sensor is actually quite large. So in terms of the solution, software happens to be the most important part of this project overall. And the cube tracking utilized open source computer vision library called OpenCV to track the cube in real time. And the microcontroller, which is the Node MCU ESP8266, is running a special version of Python called MicroPython. What this Python distribution is, it's basically a very stripped version of Python that basically allows it to run on literally about anything, because the microcontroller itself has like 8 megabytes of RAM, and it still runs really fast. So it's just a very stripped version of the normal Python that we see on our laptops today. And what it's currently running is a socket script, which all it does is it opens a network port and allows it to communicate to another device. In this case, it is capturing pressure values and sending those pressure values over the network to another computer, which is then looking through the values and putting them on a plot to visualize it, to make it easier to figure out where in particular the tumor could be located or where there's an anomaly. And these charts are usually given in the form that is understandable to both the layperson and the medical professional, due to the fact that since you track the values of the cube in X, Y, and Z coordinates, you have almost a 3D rendering of what the organ looks like and where the tumor is. So that almost replicates what a CT scan or an MRI scan can give you in terms of detail. And since the pressure sensor is very fine, it only focuses on one specific small portion of points. So we use another algorithm called a Gaussian process in order to figure out the parts of the organ with the least amount of confidence and sample those. So we sample the least number of points and still get the most accurate data. And again, we use outlier detection to report abnormal values. On the left-hand bottom side, you can see the different interaction between a client and a server, which is mainly what was used in order to create this algorithm between communicating with the server and the laptop. And on the right-hand side, you can see the pressure, how a pressure sensor works. In this case, I had to make my own pressure sensor using the Velostat. Basically, what it means is that you're plotting the different amounts of voltage based on the amounts of pressure, and you can kind of figure out the line of best fit, and using that, you can figure out how much pressure is at a certain point. So on the right-hand side here, you can actually see the cube and what the camera thought the position was. So it figured out the origin of the cube was actually in the middle of this cube here. Again, this is a 2D image, so visualizing depth might be kind of difficult, but this is exactly what the camera sees and knows the origin of the cube and the orientation as well. On the right-hand side, you can see the Gaussian process, where you can see the ending is blue, and that's the area of least confidence. So you'd sample that point again to make it so that you'd sample the least number of points, but still have the most accurate and cover a large range of data. On the left-hand side here, you can see the anomaly detection that I've implemented. In this case, you're basically seeing this cluster of points that are of different values in terms of pressure that are being categorized as potential tumor or something that's wrong in comparison to the rest of the values here. On the right-hand side, you can see the graph I was mentioning earlier, where you would have We basically plot all the values of the different organ, and using the confidence intervals, you can figure out that these areas and these areas, the outside has the least amount of pressure, and some areas which are blacker have more data that could be sampled or has an anomaly in it. On the left-hand side here, you can see the different heat map texturing. So in this model, you can see the tumor, which is a plastic disc in this case, was inserted, and you can see how the algorithm categorizes based on the pressure values. On the bottom, you can see the same different points sampled, and you can get the overall outline of the organ itself, along with the individual points of tumors. Right-hand side is just a picture of a heat map that, in relativity, shows a particular area of interest that might require further imaging. So in terms of the overall solution, these were the metrics collected. So it had an accuracy of almost 97%, which means it's almost clinically accurate. Although it's not currently being tested, it will be tested in the future in about a few months, where we have trials running on specific patients. So the software could be optimized in the future to detect tumors faster and run entirely on one device, and to shoot out small magnetic waves to show info about more invasive tumors. As you can see here, we can detect all the way up to in situ cancers, but invasive cancers get very difficult because they have more roots underneath, which I can't fully detect. So in conclusion, the goal is to change the way tumors are currently diagnosed and revolutionize the way they work by eliminating the cost and time constraints. And it can be used it can be distributed and used in underdeveloped countries to detect tumors in their earliest occurrences. And I would like to thank my family and my mentor, Aaron Ray, and as well the helpful communities at Stack Overflow and Reddit. This project would not be possible without all of you, and thank you so much. I'm now open to question and answer.
Thank you. Great presentation. You mentioned that you hope to carry out your work soon in actual trials. I was
Yes.
just kind of curious.
So I actually have been talking. So there was an implementation of this project a bit earlier, but this implementation was a very bare implementation that didn't fully function. It was an attempt, but it was an attempt by some researchers and some professors at Carnegie Mellon, and I was able to find that research, and I found out a way. I found out all the mistakes they made during their investigation. I was able to dissect it, pick it apart, make it better, basically recreate what they did in a better way that was actually functional as opposed to what they had. And I was collaborating with them throughout the extent of this project to figure out different forms of implementation and how to test it on actual patients. So that was currently what was happening now. So currently the project is still being assembled. Not all of it's done yet. We're still working out some final things, but we hope to get it tested before November. So have it tested on actual patients, and preferably, if possible, even go forward with a clinical trial.
That's amazing. We have a big picture question here. As a preventative tool, do you imagine this intervention will be limited to at-risk patients, or once implemented, become a regular part of annual wellness routines?
It actually will become a part of annual wellness routines and interventions, because currently MRI scans are very unaffordable and out of range for many patients, simply because healthcare is not free in America. I don't know if it ever will be free. And they cost $6,000 for a scan. And a scan like this with the pressure sensing costs around $15 for the product alone. And it would cost like $2 to sample. So it would not even take that much money, and it's much faster since the results are charted basically in real time. And currently the way that tumors are felt for in terms of the body are like the tissue is palpated, and that does work, but palpation usually isn't the best way in order to quantitatively define where a tumor is. You can say there is a hard substance somewhere in this region, but with this particular device, you could say at this exact point, there is a tumor 3 centimeters down beneath the skin. So in the future, I plan to hope to make it become part of annual wellness checkups and routines.
Amazing, thank you. Alrighty. Well, if there are no other last minute questions, I just wanted to thank all the presenters today for all the hard work on your projects. You guys have done some really amazing, cool things. And a huge thank you to all the audience members for coming to support these students' presentations. We at Polygence are incredibly proud of all your achievements, and I've got to say, after all the sessions I've moderated today, I am thoroughly impressed by everything you guys have been able to accomplish, and we're so glad that we got to cheer you on and celebrate you on the stage today. This, I believe, is one of the last sessions. I'm not sure if there are any other small events going on, but you're more than welcome to check them out and keep browsing through the reception button on this platform. And other than that, thank you guys again so much for your hard work. Great job today. And everyone else, have a great rest of your evening or day, wherever you may be.